Nucleic acids, a very important chapter from the perspective of CSIR NET Life Science. Today, viewers of Biotecnika, I am here with all the details that you need to know for this exam, that is nucleic acid chemistry and biology. This belongs to the unit Molecules and their interaction relevant to biology from Unit 1. Subunit B and H will be covered here. Let's dive in. As we know, nucleic acids are linear polymers of nucleotides. Nucleotides are the building blocks and nucleic acids are the polymers of nucleotide. Now, what is a nucleotide? In fact, there is another term which is nucleoside. Nucleotide is a component formed of three different elements, that is phosphate group, the ribose sugar, or what is also called as pentose sugar because it is a 5-carbon sugar molecule, carbohydrate. And apart from that, you have the nitrogenous base. The name is because it has a lot of nitrogen content, and also they have amino groups, that's why the basic nature. And nucleoside contains only the ribose sugar and the base. It doesn't have the phosphate group. This will be only the nucleoside part. We have two different types of nucleic acids, which is the DNA and RNA. The difference between them is in the ribose sugar. Ribose sugar numbering starts from this carbon, which is 1 dashed. The dash comes because purine and pyrimidine also have numbering which starts with 1, 2, 3, 4 like that. And that's why the ribose sugar numbering, just to differentiate it from the purine and pyrimidine, they have a dash attached with it. 1 dash, 2 dash, 3 dash, 4 dash, and 5 dashed. The difference between DNA and RNA is in the 2 dashed OH group, which in case of DNA, it is just hydrogen, where the oxygen is missing from here. That is why the name deoxy. D means removal of the oxygen. Whereas in RNA, you will have as it is the OH group. The different DNA and RNA nucleobases, they are mainly categorized into purines and pyrimidines, the nitrogenous bases. Purines are of two types, which is called as adenine and guanine. Pyrimidines are of three types, that is cytosine, uracil, and thymine. You will have to know the structure of all of them, but it is not at all difficult if you once understand the numbering of the nitrogenous bases. Purine has 2 rings in it, you can remember like that. And the position of nitrogens I have circled here, as you can see. In one of the ring, it is a 5-membered ring, another ring is 6-membered. In the 5-membered ring, the nitrogen positions are at the top and bottom. In the 6-membered ring, the position of nitrogen is in the side and at the bottom. Now, whenever you are supposed to number, and anyways pyrimidine is only one ring, which is of 6-membered ring, the position of the nitrogen is in the side and at the bottom. Now, whenever you start numbering for the nitrogenous bases, remember for purines you are supposed to start from the side nitrogen, and for pyrimidine you can start from the bottom nitrogen. And you have to move to the nearest nitrogen with the numbering. So, for example, pyrimidine, the nearest nitrogen is this one. So, from 1 you can go to 2, 3, 4, 5, 6. That is how you will number. For purines, you start with the side nitrogen and you can proceed towards the nearest nitrogen, which is this one. So, 1, 2, 3, 4, 5, 6. After completing 6, you can come directly to 7 and then 8 and 9 like this. Now, how are you supposed to remember these nitrogenous bases, different types? You can remember the position of the functional group. For example, in adenine, you have an amino group which is present at the 6th position. And because adenine is a type of purine, that is why you can name this as 6-aminopurine. This is how you can remember. In guanine, you have the position of amino group at 2nd. And at the 6th position, you have a oxo group. So, accordingly, it will be 2 amino and 6 oxoguanine. Likewise, you can remember for cytosine, uracil, and thymine. For all of them, a common factor is you will have oxygen at the 2nd position with a double bond. The difference between uracil and cytosine is that at the 4th position, you have an amino group for cytosine, but for uracil you have a oxygen group. And the difference between uracil and thymine is only a methyl. That is why thymine is sometimes called as a methylated uracil. Okay. The only difference between uracil and thymine is that thymine is sometimes called as the methylated uracil. There will be other modifications of the DNA bases, DNA and RNA nucleobases, which you will be seeing in different RNA structures like tRNA, or in the different DNA mutations of the bases that we have. However, that is beyond the scope of this lecture discussion. This you can find it in unit 3 topics, under 3 topics, and under unit 8, that is genetics topics. So all ring atoms, these are all aromatic rings. That is why these all will be planar structures. Whenever it comes to nucleic acids in CSIR, they will ask very topic related, that means term related. You have to focus on the different terms and you have to understand the keywords. Okay, you have to remember and recollect the keywords. So while making notes, summary notes, you can underline the keywords here. Here right now in aromatic ring, the keyword is that they are planar structures. Now planar structures, they can ask you whether DNA is chiral or not, or whether DNA is planar or not, and what contributes to the planarity. So here the answer is the bases contribute to the planarity of the DNA molecule, whereas the ribose sugars, we know these are chiral molecules, ribose sugars, so they contribute to the chirality of DNA molecules. Okay. Now coming to the bases, different types, that is nucleoside and nucleotide. As I said, nucleosides are without phosphate group. So the different types, if it is ribose nucleoside, it will be adenosine with the adenine, and if it is a DNA nucleoside for adenine, it will be deoxyadenosine. When you have nucleotide, all you have to do is you have to attach a phosphate group, and that is how it will become either monophosphate. So accordingly it will be AMP or ADP or ATP. And in case it is DNA, you have to attach the letter D, small d to it. Okay. Going into some more details, as we know the sugar molecules, that is ribose in RNA and deoxyribose in DNA, these predominantly exist in the cyclic form. If we find this linear structure, what we draw generally, but generally these groups, they are found in cyclic form, something like this. Okay. Now anyways, if you are aware that carbohydrates, what is most abundant in nature, they are of D form. Okay, like how amino acids are always found in L form, the carbohydrates are mostly found in D form, and this is called as a D ribose. But you can have a ring form in which there arises one more anomeric carbon, which in this case C1 is not a chiral carbon, rest all carbons are except this one. But when the cyclic form happens, even this number 1 carbon gets anomeric in nature. Okay, that is chiral in nature. So this is called as an anomer, and for this the different orientations of the OH group determines the different types of anomer, that is alpha anomer and beta anomer. Okay. Now if the OH group is in the same plane as of the 5th carbon, then it will be called as a beta anomer, and if the OH group happens to be down, then it will be an alpha anomer. Since OH group is present here, that is why this is a beta anomer and it is a D sugar, and this is called as ribofuranose. These complete terms may be asked in question, like what is the ribose sugar in DNA or what is the ribose sugar in RNA, and they will change these small things, right? So they can make it alpha or they can make it L, they can change the number. So where the deoxy is happening, it is at the 2' position. So you must know all this. Furan is coming from the word the ring structure. In this case, the ring structure is a 5-membered ring in which 1 is oxygen and the 4 are carbon. Such a ring is called as a furan ring. And in case you are having a pyran ring, you will have a 6-membered ring. So since this is a 5-membered ring, that's why we can call it deoxyribofuranose. Okay. Now, you have to know some conformation changes of the sugar molecules and the different bases, their conformations, and also what are the type of bonds which you find in nucleotides. So first we will look at the sugar ring puckering. Now what is this? We know that carbon, it has a valency of 4 and carbon has a tetrahedral structure. So by adopting different, to be in a stable form, carbon takes up different conformations by taking the puckering conformation. So sugar rings, they can alter the relative orientation with the phosphate backbone, okay, and also the nucleobase and also the ribonucleotide. So you can see this ribose sugar, 2 different conformations are shown on screen. The first one, you have the number 1 carbon which is attached with the base, and then the second carbon in the sugar, it is slightly risen up from the plane. That whatever will be the plane group of the planar plane angle of the sugar, from there the C2 dash is slightly towards the inside. So this particular conformation is called as endo. If you see like that, it is with regard to the C5 dash carbon and the base. So if the particular atom in concern, it is towards that C5 dash and base, then it is in an endo conformation. Okay. In comparison, you can check this diagram where the C2 dash, you will see it is away from the 5th carbon and the base. So if it is out of the plane of C5 and the base, it is called as a exo conformation. Now which atom I am referring to? I am referring to C2 dash. So in this it is C2 dash is endo, and in this C2 dash is exo, right? But if C2 dash is endo, then the near one, that is C3 dash, it has to be exo. And in case the C2 dash is exo, then C3 dash has to be endo. Both cannot be endo or both cannot be exo conformation at the same time. Based on this also questions come, so you should be very much clear with this concept. Now question is, which puckering will be more favorable? So sugar pucker and the different pp distance, that is between the 2 phosphate group, what is the difference? As I said that the whenever you have the C2 dashed endo, at that time the C3 dashed will be outside. So you can see when the C2 dashed is endo, the C3 dashed and the phosphate, the angle between them is, the distance between them is more in this case. If you can follow in this diagram also, when C2 dashed is endo, the distance between the 3 dashed carbon and the phosphate group, it is more. But when C2 dashed is exo, C3 dash has to be endo. In that case, the distance between the phosphate group is very less. So this might lead to steric clash, because of which this is not so much favored. More favored is where C2 is inside, that is, it is in endo conformation. Okay. So remember, this is a more favored one. C2 dash endo is a more favored one. Now what about the bond between sugar and nitrogenous base? So the bond between the sugar and nitrogenous base is a glycosidic linkage. It is a misconception that glycosidic linkage can only be formed in between 2 sugars. So all you need is just a sugar, as you can see, and whenever the base is connected with it, this C dashed OH group and from the base H group is removed with the loss of water molecule, which results in the formation of a glycosidic bond. Okay. And then we have the base also, the nitrogenous base along with the phosphate group. Now another thing is how they are connected. If you see for the purines, the connection with the sugar is with the N9. Okay. And remember, in case of pyrimidine, there is only one nitrogen which comes in the bottom, and that is the 6th nitrogen. So for pyrimidines, the connection, which is the first nitrogen, okay, because we start the numbering of pyrimidines from the first. And for pyrimidines, there is only one nitrogen which comes at the bottom, and that is the number 1 nitrogen where we start the numbering. So the C1 dash, the sugar is connected with the first nitrogen in case of pyrimidines, and in case of purines it is connected with N9. Okay. So this is the connection with the pyrimidines, as you can see N1 and the C1 dashed. So this type of questions are also asked. C1 dashed and N1 are connected together, whereas here C1 dashed and N9 are connected together. Okay. Next are the glycosidic bond conformations. So glycosidic bond means the bond that we just saw, the bond between the sugar and the base. This bond can exist in two different conformations. As in what? The C1 dashed and the O4 dashed. That means this one and this one. Whereas the N9 and C4, that means the two nitrogen, if they are pointing towards the same direction, then this arrangement is called as syn conformation. This arrangement of the glycosidic bond, it is syn conformation. If the C1 dashed and O4 are pointing in one direction, whereas the two nitrogens are pointing in opposite direction, then it is an anti-conformation. So this can be possible even for pyrimidines also, but for pyrimidines the reference is this oxygen. I told you all pyrimidines have the oxygen at the second position. So if this are pointing in this direction, in one direction, and the nitrogen and oxygen are pointing in the other direction, then this is the anti-conformation for pyrimidines. Okay. Now there are different forms of DNA where these different groups have different glycosidic bond conformations. So you must remember and understand what these are. These are defined based on the torsion angles of the glycosidic bond. Right. Moving on, the bond between the sugar and the phosphate. As we all know, if a phosphate, if we are talking about a single nitrogenous triphosphate, then we have the sugar, we have the base, and then we have three phosphates connected to it. Okay. So the first phosphate is an alpha phosphate, then is a beta phosphate, then is a gamma phosphate, and the bond between the sugar and the phosphate, the single phosphate connected to a single sugar, it is called as phosphomonoester bond. Okay, it is an ester bond, but it is a monoester because it is connected to only a single sugar. But in case the phosphate group is attached to two sugars at both ends, then it becomes a phosphodiester. That means two ester groups are there, one at each end, and this is what happens. Nucleotides are linked by phosphodiester bonds. The ends are referred to as 5' end and 3' end because at the 5' end you have a phosphate group attached, that's why it is 5' phosphate. At the 3' end you have a free OH group, the terminal one, that's why this is the free 3' OH end. The strands will have a direction. We always write 5' to 3'. How we write for amino acids from N to C terminal, here also we will write like that. In case you are having a double-stranded nucleic acid, then the strand will run anti-parallel to each other. And we all know that DNA is negatively charged molecule because of this phosphate group which is present in the backbone. Even after bonding with sugar, this is the only molecule which can still carry a negative charge, and this is a very important property which is significant in the movement of nucleic acids in electrophoretic field. Next is purines always binds with a pyrimidine. That means what is the type of bonding when we have a interaction of two strands of nucleic acids. So we know that purine will always bind with a pyrimidine, like thymine or uracil will always bind with adenine, and cytosine will always bind with guanine. That is the CG bond which are connected by 3 hydrogen bonds, and then we have the AT bond which are connected by 2 hydrogen bonds. Okay. Now here, as we all can see, the hydrogen bond, it is characterized by the donor and the acceptor, right? So here NH group is acting as the donor and oxygen or nitrogen is acting as the acceptor, right? Now guanine is the only molecule which has 2 donors from attached to it. Okay, rest all have only one donor. That is why guanine has the maximum possibility of hydrogen bonding. Very small and minute details are present in this structure. Ensure you don't miss any of them and you write it down, because in some question, in some sentence, these questions, sentences and details might be required. Okay. Apart from Watson-Crick base pairing, so this what I showed you was a Watson-Crick base pairing in their model, where the N1 of the purine is connected with the N3 of the pyrimidine. Yes, you have to know the atom numbers also. That is why we discuss the numbering. Watson-Crick assumes that both the purine and pyrimidine are in anti-conformation. Okay. But there is also another type of pairing which is called as Hoogstin pairing, in which the nitrogen of purine, it is not N1 but it is N7. So you can see that this is rotated this side, isn't it? So the conformation is different over here. This is what is N7 and it is connected with N3 of pyrimidine. That is same, it's just that this has been rotated. So where purine is in syn form and the pyrimidine is in anti form, this is what forms the Hoogstin pairing. Okay, and triple pairing is possible, where one can be with Watson-Crick pairing and one can be with Hoogstin pairing, right? So based on this, we have Chargaff's rule. In all cellular DNAs, regardless of the species, A will be equal to T, that means the number of purines will be equal to number of pyrimidines, and G will be equal to C, provided it is a double-stranded DNA. From this relationship, it follows that the sum of the purine residues will equal the sum of the pyrimidine residues. That means A and G, the total number of purines, will be equal to T and C, which is the total number of pyrimidines. A by T will be 1 and G by C will be 1. AT base pair, it doesn't mean that AT base pair will be equal to GC base pair. No, in some DNA, the GC content can be more. And in fact, since those are connected by 3 hydrogen bonds, it will need more energy to denature the DNA double strand. Denature means separate the 2 strands from each other. Whereas in AT base pair, it will need less energy. So some questions are based on this property of Chargaff's rule also. A plus T by G by C ratio, typically denoted percentage GC. How much is the GC content? It varies from DNA to DNA and from organism to organism. The composition of DNA varies from species to species, but A plus T by G plus C, it is constant for a species. For a species, it will be constant. Next topic is polymorphism of DNA double helix. Double helix DNA, it exists in different forms. So we have the native DNA duplexes in fibers, which exist usually in one of the 3 well-known forms, which is ABC form, and these different forms will change based on the relative humidity, how much is the humid content, type of the cations which is present in the environment, and how much salt it is able to retain. For example, if you have the Watson and Crick model, which is the BDNA model, if the conditions are more dehydrated, for example, if you have here this DNA, if this DNA is in less humid condition where it will become dehydrated, the DNA molecule becomes little bit shorter. Okay, and that is where the diameter, if the DNA becomes shorter, the diameter will become longer. So it becomes a short and thick form of a DNA. It's just that it is another form of it. This is only what is called as aDNA. Okay. So these are all different secondary structures of DNA where you can get different alternative forms. Like B form will be having duplex structure proposed by Watson and Crick. It is a standard structure for DNA molecules. A form, it forms in low humidity. The DNA molecule will take the A form. A form is wider and shorter with a shorter, more compact helical structure than the B form helix. Whereas Z form, it is a special type which is only adapted by a specific type of sequence, that is alternating cytosine and guanine. So these are generally adopted by stretches of short oligonucleotide. Remember that both B and A forms are right-handed helix, whereas Z form is a left-handed double helix in which the backbone phosphate will be zigzag manner. So this is how the different forms of ABZ DNA are. As you can see, Z is a more elongated form where the diameter will be comparatively lesser. Okay. Some more differences will be there, like B DNA has a wide major groove, whereas A DNA has a narrow major groove. The major groove will be narrower in this case. And in fact, Z DNA, it doesn't have any major groove. The strands are so flat that you may have a narrow minor groove, but then major groove is completely absent. Okay. So these are different features and dimensions of the B DNA helix. As I told you, B DNA is the most relaxed form of DNA. That is why the sugars are in the 2' endo conformation. No steric hindrance. The 3' group and the phosphate group are at much distance, avoiding steric hindrance. The bases are in anti-conformation. Again, this is favorable because the base is away from the sugar phosphate backbone. So this is also in a relaxed form. The helical pitch. Pitch refers to the rise per turn. A turn in a helix refers to as 360-degree rotation. The length of the helix of one turn, it is 34 Armstrong. You can also refer it in nanometer. And the distance between two bases, it will be calculated based on how many bases are there in one turn. So in BDNA, roughly there are 10.4 bases per helix turn. So to find the rise per base pair, which is the distance in the helix between two bases, you have to divide the pitch divided by the number of bases, which is 10.4. So the rise per turn will be 34.6 degrees. Okay. Whereas in ADNA, if you see, the sugars are in the 3' endo conformation. I said 3' endo is not so favorable, but whenever it is required, like in less humid conditions, the DNA will become dehydrated, so it will be forced to be in this particular form. So here sugars are in 3' endo conformation. However, bases still remain in the anti-conformation. This DNA is slightly wider than the B DNA. So where B DNA has 20 Armstrong diameter, which is 2 nanometer, here the diameter is slightly more, which is 25.5 Armstrong. Since the DNA is shrink little bit, more bases will be accommodated in one turn. That is why here more bases, 11 bases will be present per helix turn, and the helical pitch here will be shorter than B DNA, which was 34 Armstrong. Here it is 25.3 Armstrong. Also notice that the bases are pushed more towards the center, whereas in BDNA the center part will be comparatively empty because the bases will be farther away towards the periphery. Okay. Z form is a special type. This is a left-handed helix, and in BDNA and ADNA the helix backbone goes into a straight line. But this is the only DNA where the helix will be going in a zigzag line. And here the bases will alternate between anti and syn conformation. That is between the pyrimidines and the purines. This is a slightly elongated DNA. That is why you will have 12 bases per turn. And the helical pitch over here will be more. Here the one turn is 45.6 Armstrong. However, it is a more thinner type of helix. That's why the diameter is only 18.4. But the most important thing to form a Z DNA is that you have alternating GC sequences here, and that too there is a specific requirement where you can see that the C sugar, it is in endo, 2' endo form, and base is in anti form, whereas the guanine sugar, it is in 3' endo form and the base is in syn form. Because of the different conformations of the C and G, they can become alternate to each other, which leads to a zigzag in the backbone. You can refer to the Biotecnica's concept table book, which has all the details of the differences between ABZ DNA, which are very helpful to revise during exams. The last point before I go to it, one more important point is that B-type duplex is not possible for RNA. Whenever we have this RNA, the B RNA will not be possible because in RNA the 2' oxygen is present, and this oxygen makes a steric clash, because in B DNA we know the 2 dashed is present in endo form. So in endo form, because of this oxygen, there will be a steric clash. So B type RNA is not possible. Accordingly, keep it in mind while solving questions. And the different forms of the DNA and RNA, the nucleic acids, you can have some non-helical secondary structures as well. As we saw the helical secondary structures, the different polymorphic forms, you can have non-helical forms also, like something which is called as a palindrome. Now what is it? If you have the palindromic sequences on one of the strand and the palindromic sequence on the other strand, then those two will come together in the form of a hairpin because of complementary binding. So such kind of sequence based questions are asked regarding mRNA, not mRNA, rRNA and tRNA in CSIR paper, where based on the sequence, even for microRNA, they will ask you to show which of these sequences might form a miRNA or a tRNA. So all you have to look is for complementary base pairing between the palindromic sequences, which can lead to the formation of a hairpin structure, or if it is in both strands, which will form a cruciform structure. Okay. So inverted repeat can lead to a loop formation. That's why you should look for the sequence carefully. The tertiary structure of DNA is consisting of supercoils, where you have the double-stranded DNA, where one strand is coiled with the other. So coiling is done. Now if this supercoils means you have more coils with this, then it is called an increasing degree of supercoiling. This can be of two types: underwound, which is negatively supercoiled, or overwound, which is positively supercoiled. So the negative supercoil will twist the DNA about its axis in the opposite direction from the clockwise turn of the right-handed helix. This will be underwound because it is twisting in the same direction, whereas the positive supercoil will twist in the same direction as the turns of the right-handed supercoil. So here the turns will be overwound. So helix would be wounded up more tightly. Okay. So you can imagine like a telephone coil where already it is in the helical form, but if you keep on turning it even more, then it will lead to supercoils like this. This is generally present in plasmids, that is covalently closed circular DNA, and even in eukaryotic DNA we can see in the formation of a replication fork, where ahead of the replication fork portions of DNA will be positively supercoiled. That is why we require the topoisomerases or gyrase to change this nature of supercoiling. Okay. So this is a diagram where you can see if the strands are overwound with each other, then it leads to positive supercoil, which is a left-handed supercoiling nature. And if the strands are underwound, then it will lead to a negative supercoil. I hope this lecture was beneficial to you. Let me know in the comment section. Thank you.